So our final speaker, last but not least, is Zhang from another MD PhD student from Yale University, who will talk about additional data on the neuroinvasion of the SARS-CoV-2 virus using data from organoids and post-mortem tissue. So thank you, Tse. Looking forward to your talk.
All right. Thanks a lot. Hi, everyone. My name is Zhang, and I'm an MD PhD student at the Yale School of Medicine. And today I'll present our work on the neuroinvasion of SARS-CoV-2 in human brain organoids and post-mortem tissue. So there's been many papers about the neurological findings of COVID-19, but I want to briefly talk about a study that was published about 2 weeks ago that examined neurological manifestations in 509 patients admitted with confirmed COVID-19 in the Chicago area. The study found that neurosymptoms were present in about 60% of hospitalized COVID-19 patients and about 80% at any time over the course of the disease. And these were some of the symptoms that were reported in encephalopathy. It comprised about 32% of the symptoms. Encephalopathy is a term that describes an altered mental status in the patient, such as a decreased level of consciousness, problems with attention and concentration, etc. And the authors found that in the encephalopathic patients, there was almost a 3-fold increase in the length of the hospital stay, increase in the percentage of 30-day mortality, and about 32% of the patients that were discharged were unable to look after their own affairs without assistance, compared to 89% in non-encephalopathic patients. So the takeaway message is that the neurological complications of the infection are clinically important. They can be frequent, long-lasting, and can impair function. The major question is, how does COVID-19 cause the neurological symptoms? And 2 potential hypotheses includes a direct infection of the neural tissues or immune system-mediated neuroinflammation. We decided to focus on the possibility of direct viral infection and ask whether SARS-CoV-2 can directly infect the brain. So we utilized 3 model systems to determine the neuroinvasive potential of SARS-CoV-2, the mouse brain, human brain organoids, and human post-mortem brain tissue from COVID-19 patients. And for this talk, I'll just focus on the brain organoid and post-mortem brain models. So we first infected 60-day brain organoids and fixed them at 96 hours post-infection for analysis. And using the SARS-CoV-2 nucleic acid antibody, we identified SARS-CoV-2 positive radioglia, that's marked by SOX2, and also neurons marked by MAP2. And here's a wide view of our SARS-CoV-2 infection, our organoids showing infection mostly on the outer regions of the organoid, and they're mostly composed of neurons, these outer regions. And furthermore, we found that these infected cells were positive for 2 markers of cortical neurons, CTIP2 and TBR1, indicating that SARS-CoV-2 can infect cortical neurons in organoids. Here we stained for TUNEL, which is a late-stage cell death marker, and found that MAP2 positive neuronal regions infected by SARS-CoV-2 had increased TUNEL staining here, shown in the green, indicating neuronal cell death. But what was interesting was that the SARS-CoV-2 infected cells were mainly TUNEL negative, while the neighboring non-infected cells were TUNEL positive. Moreover, within a region, the density of the TUNEL positive cells positively correlated with the density of SARS-CoV-2 infected cells. And here in this yellow box, this is an infected region showing infected cells in red, TUNEL positive cells in green, versus a white box indicating a non-infected region with less dying cells. So we ultimately hypothesized that the SARS-CoV-2 infected cells can somehow promote the death of the neighboring cells, neighboring non-infected cells. So to uncover a mechanism for this hypothesis, we used 10x single-cell sequencing to analyze the transcriptome of SARS-CoV-2 organoids, infected organoids at 96 hours post-infection, and sequencing detected viral RNA present in many single cells in the infected organoids versus no detection in the mock. And then next, within infected organoids only, we compared the transcriptome of SARS-CoV-2 positive cells versus the neighboring non-infected cells. And in SARS-CoV-2 positive cells, we found upregulated pathways, including viral transcription, electron transport chain activity, a response to hyperoxia, suggesting a hypermetabolic state in these infected cells. In contrast, in the SARS-CoV-2 negative cells, we found upregulated pathways corresponding to cellular response to hypoxia, increased cell death, indicating that in response to hypoxia. So our interpretation of the sequencing data is that SARS-CoV-2 infection leads to a hypermetabolic state and promotes a hypoxic state in the surrounding non-infected cells. We confirmed that in SARS-CoV-2 infected organoids, there was an increase in cells positive for HIF-1 alpha, a marker for hypoxia. And then we also treated organoids with pimonidazole compound, which is another known hypoxia marker. And we found an increase in the pimonidazole intensity in the SARS-CoV-2 infected regions compared to the non-infected area. So altogether, this suggests that SARS-CoV-2 infection is potentially harmful to the surrounding cells by promoting local hypoxia. Moving on, it's known that ACE2 is a receptor for SARS-CoV-2 and facilitates the entry of the virus into host cells. So then we wondered whether ACE2 was also expressed in the neural tissues. So we performed immunostaining for ACE2 and isotype control in the brain organoids, and we found that ACE2 was widely expressed throughout the organoid here, shown in white, and strongly colocalized with MAP2 staining, indicating that it is expressed in organoid neurons. And then here we did ACE2 staining in adult human cerebral cortex tissue and found that ACE2 was present in the new and labeled mature neurons. So neurons from both the brain organoid and human post-mortem tissue express ACE2. Now we asked whether ACE2 was required for SARS-CoV-2 infection in brain organoids. And for this experiment, we incubated the organoids with ACE2 antibody to block the organoid ACE2 receptors or isotype antibody for 1 hour before infecting the organoids with SARS-CoV-2. And then organoids were analyzed at 96 hours post-infection. And we found a significant reduction in SARS-CoV-2 infection in organoids pre-treated with the ACE2 blocking antibody. So this suggests that the SARS-CoV-2 infection in brain organoids is ACE2 dependent. Lastly, I want to show some data from human post-mortem brain tissue of 2 COVID-19 patients. These data were analyzed by neuropathologists at Yale. Patient 1 was a 63-year-old male with a past medical history of hypertension on a ventilator 10 days after symptom onset. And then we used a well-established spike antibody against SARS-CoV-2 protein. We detected positive staining in the frontal lobe of the brain, as shown here, as well as in blood vessels. And these are cortical neurons. And then patient 2 was a 49-year-old female on a ventilator 3 days after symptom onset and was diagnosed with encephalopathy on hospital admission. The past medical history had many comorbidities. And what we found here was that in the caudate nucleus, we found ischemic regions located left of the dashed black line here versus a non-ischemic area here on the right. And in these ischemic regions, we found SARS-CoV-2 positive neurons, as well as microvasculature. But it's important to note that we cannot determine the causality of the SARS-CoV-2 infection in ischemic areas. So in conclusion, SARS-CoV-2 can infect cortical neurons and brain organoids. It can promote local hypoxia and cell death in the neighboring cells. It can infect brain organoids in an ACE2-dependent manner, and it can be detected in neurons and microvasculature and COVID-19 patient post-mortem tissue and is associated with ischemic areas. So altogether, these results provide evidence for the direct neuroinvasion of SARS-CoV-2 in the CNS. This work wouldn't have been possible without the help of our many collaborators, and I want to especially thank my PI Kaya, as well as Akiko Iwasaki and Eric Song, who you guys may have heard from in the ISSCR presentations. I want to give thanks to the families of the patients for tissue donation and the New York Stem Cell Foundation for providing an IPS line used in this study. Thank you.
Thank you so much, Joe. Yeah. That was really beautiful work. And I was curious, I know that you had a little bit more that you've kept for the poster with your mouse work. Do you want to say a word or two about that as a plug for your poster presentation this afternoon?
Yeah, sure. So we use like a K18 mouse line that expresses the human ACE2 gene from the K18 promoter. And we found widespread infection of neurons with positive staining for the SARS-CoV-2 virus in the mouse. And also in the mouse, we detected changes in the vasculature within the SARS-CoV-2 infected regions. In fact, in the SARS-CoV-2 infected regions, we found sort of decreased vasculature in that area versus the normal mouse. So that maybe suggests that perhaps the vasculature was like diverted to a more metabolic, highly metabolic area versus the SARS-CoV-2 infected area.
Okay.
Yeah.
All right, great. Actually, that relates to a question that came up just now. Could that maybe represent a primarily vascular disease rather than primary neuron infection, what you're seeing? That's a great question. It's still, I guess, controversial whether
neurological effects are due to maybe like an infection of the vasculature versus the infection of the neuron. So that's something that we'll definitely look into in the future. Yeah.
Okay, great. Excellent. Well, thank you so much for sharing that data with us and to all of our presenters for a really great talk.